The overall goal of this procedure is to visualize purified protein samples by negative stain electron microscopy. This is accomplished by first preparing the electron microscopy grids that will support the sample. The second step of the procedure is to prepare the negative stain solution. Next, the sample is applied onto the electron microscopy grid and negative stained. Lastly, the EM grid is loaded into an electron microscope and images are recorded under appropriate optical conditions. Conventional bright field TEM images are high contrast and sharply detailed, showing single macromolecules scattered over a featureless background.
Negative stain electron microscopy is a simple method of visualizing your favorite protein. It can be used as a routine assay to examine your purified protein, either for structural analysis or other biochemical and biophysical assays. Here we demonstrate how to prepare a negatively stained sample for electron microscopy. Visual demonstration of this method is helpful, as some of the steps are difficult to learn from text descriptions and figures.
Start by using a glass pipette to place a single drop of collodion into a large beaker of water on the water's surface. As the collodion spreads, it may capture dust from the water's surface. Remove and replace a dusty film of collodion plastic with another drop. Next, place between 50 and 100 EM grids one by one onto the floating plastic film, shiny side down. Select 400 mesh grids for routine negative stain, and select 200 mesh grids for collecting tilt pair images. Select a piece of slow absorbency unwrinkled paper that is slightly larger than the area of the grids. Place the paper on top of